Myself, Mallanagoud G, Assistant Professor of Zoology, Department of Zoology, Veerashaiva College, Ballari. Our college belongs to BSK University, Ballari. Today, I am going to explain about the bioenergetics. First, I would like to explain the introduction regarding bioenergetics. Bioenergetics is the biology of energy flow, energy transformation, and energy exchanges within the living systems and between living things and their environments. Sun is the ultimate primary source of energy for all living organisms. The green plants convert the solar energy into chemical energy, that is, they can manufacture their energy rich carbohydrates by means of photosynthesis. Therefore, they are called autotrophs. All other organisms on this planet are heterotrophs as they obtain their energy by consuming other organisms. The energy present in the living organism, which is used for all their life activities, as well as the energy content of the products derived from the living organisms, that is wood, biogas, etc., is called bioenergy. The biological process like photosynthesis, cellular respiration, and cellular work play a major role in the energy transformation. The plants convert light energy into the chemical energy. In the cells, the chemical energy carrier is ATP. ATP is energy molecule and energy is stored in the phosphate bonds. The energy present in ATP is used for the fixation of CO2 into carbohydrates. All body cells transform the energy of food molecule into energy-rich ATP molecule. This process is called cellular respiration. ATP molecules are unique energy sources of living system. ATP is called the energy currency of the cell. In a biological system, energy yielding and energy consuming reactions occurs. Therefore, the energy transduction in a living system is strictly in conformity with the laws of thermodynamics. Energy flow, transformation, and energy exchanges within living system are governed by laws of thermodynamics. According to the law of thermodynamics, energy can neither be created nor destroyed. Hence, the energy is conserved in one form or the other in this universe. Next, I would like to explain the respiration. Respiration is defined as the biological oxidation of food molecules like carbohydrates, proteins, fats, leading to the formation of utilizable energy in the form of ATP molecules. Another definition of respiration is the biological oxidation of complex organic compounds like glucose, proteins, and fats into the simpler substances like carbon dioxide and water, with the release of energy in the form of ATP molecules within the cell is called respiration. The process of respiration, that is aerobic, can be summarized in the following equation. The glucose, when it is combined with oxygen to form carbon dioxide, water, and release the energy in the form of 38 ATP molecules in aerobic respiration. The ATP has adenine, ribose, sugar, and 3 phosphate groups. The phosphate to phosphate bonds are the high energy bonds. When the phosphate bond is hydrolyzed, the chemical energy is released and the same is utilized by the cells for its life activities. Now, I would like to explain the sites of respiration. Cytoplasm and mitochondria is the site of respiration in a cell. ATP synthesis is the main principle behind the respiration. In Greek word, mitos means thread, chondron means nothing but a granule. Mitochondria is a cell organelle found in the cytoplasm of the cell. It is also regarded as powerhouse of the cell. The mitochondria was first discovered by Kolliker in the year 1850. The later term mitochondria was coined by Benda. The number of mitochondria varies from one cell to another cell. That is, number of mitochondria is depending upon the metabolic activities of the cell. Generally, the plant cells are having lesser number of mitochondria when compared to the animal cell. Sperm cells consist of 20 to 24 mitochondria, whereas sea urchin egg possess 13,000 to 15,000. Next one is where do you find the mitochondria? Mitochondria is found in all the eukaryotic cells but are absent in prokaryotic cells like bacteria and blue-green algae. Shape of the mitochondria is usually a rod shape or spherical shape or club-shaped structure. It measures about 3 to 5 microns in length and 0.5 to 1 micron in diameter. Let us we study the ultrastructure of mitochondria. Mitochondria is bounded by the double-layered membrane. Outermost membrane of the mitochondria is called outer mitochondrial membrane, and inner layer of the mitochondria is called inner mitochondrial membrane. The outer mitochondrial membrane is uniform, but an inner membrane is provided with numerous infoldings called cristae. The space present in between outer and an inner membrane is called perimitochondrial space. Here it is having 40 to 80 angstrom in wide. The fluid enclosed by inner mitochondrial membrane is called mitochondrial matrix. The matrix contains DNA, ribosomes, granules, and enzymes. The cristae provides the space for the attachment of the elementary particles called Racker's particles. Each Racker's particle has head piece, stalk piece, and base piece. Racker's particles contains many enzymes which are involved in electron transport system, oxidation, Krebs cycle to release the ATP molecules. The chemical composition of dry mitochondrion is 60% to 70% of proteins, 25 to 30% of lipids, and 9 to 10% of RNA. There are two types of respiration, that is aerobic respiration and anaerobic respiration. First, I would like to explain aerobic respiration. The definition of aerobic respiration is the process in which the complete oxidation of fuel molecules into simpler molecules like carbon dioxide and water in the presence of oxygen molecule is called aerobic respiration. Another definition of aerobic respiration is the process in which complete oxidation of food in the presence of oxygen to liberate the energy is called aerobic respiration. Then what do you mean by aerobe? Aerobes are the organisms which break down the fuel molecules completely in the presence of oxygen. Aerobic respiration occurs in higher plants and animals. It involves a stepwise breakdown of fuel molecules like glucose. Energy is released and some of the steps is used in synthesizing ATP molecules. Aerobic respiration involves three important steps. First one is the glycolysis, second one is the Krebs cycle, third one is the electron transport system. Aerobic respiration can be summarized by the following equation. When the glucose molecule is combined with oxygen to form carbon dioxide, water, and release the energy in the form of ATP molecules and heat. Here in aerobic respiration, 38 ATP molecules are released by the breakdown of one molecule of glucose. Now I will take the detailed account of the steps involved in respiration. First one is the glycolysis. Glycolysis is also known as Embden-Meyerhof pathway. Glycolysis may be defined as a series of reactions in which 6 carbon glucose molecules are converted into 3 carbon pyruvate molecules. The scheme of glycolysis was discovered by 3 German scientists called Emden, Meyerhof, and Parnas, and therefore the glycolysis is referred to as EMP pathway after the abbreviation of their last names. It occurs in the cytoplasm of the cell and does not require oxygen. Glycolysis involves the following steps. First one is the phosphorylation. In this step, glucose molecule is converted into glucose 6-phosphate in the presence of hexokinase enzyme. In this step, an ATP is used. Second step is the isomerization. The glucose 6-phosphate is converted into fructose 6-phosphate in the presence of phosphoglucose isomerase enzyme. Third step is phosphorylation. The fructose 6-phosphate gets phosphorylated to form fructose 1,6-diphosphate by utilizing another ATP molecule in the presence of phosphofructokinase enzyme. Thus, two ATP molecules are used during glycolysis. The fourth step is the cleavage. The fructose 1,6-diphosphate is splitting into two 3-carbon compounds like 3-phosphoglyceraldehyde, that is PGAL, and a molecule of DHAP, which are interconvertible. Then fifth step, phosphorylation and dehydrogenation. The 3-phosphoglyceraldehyde is oxidized to 1,3-diphosphoglyceric acid by an enzyme called dehydrogenase. A molecule of NADH and H, that is NADH2, is formed during this reaction. Each is also now oxidized, that means the removal of hydrogen or dehydrogenation. This is the schematic representation of the glycolysis, showing the stepwise breakdown of glucose molecules into pyruvic acid. Step during glycolysis is dephosphorylation. The 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid by the phosphoglycerokinase enzyme. A phosphate molecule is released and combined with ADP to form ATP. Seventh one is rearrangement. The 3-phosphoglyceric acid, that is 3-PGA, is converted into 2-phosphoglyceric acid, that is 2-PGA, due to shifting of phosphate from third carbon position to second carbon position. It occurs in the presence of a phosphoglyceromutase enzyme. Then eighth step in glycolysis is dehydration. A molecule of water is removed from each phosphoglyceric acid molecule to form phosphophenol pyruvic acid by an enzyme is called enolase. The ninth step in glycolysis is dephosphorylation. The phosphophenol pyruvic acid is converted into pyruvic acid in the presence of pyruvate kinase enzyme. In this process, phosphate group is removed and it is joined to ADP to form ATP. In glycolysis, the glucose is converted into 2-pyruvic acid. As a result, 4 ATP molecules are produced in general, but 2 ATP molecules are utilized at the beginning of the glycolysis. Here, in the end of glycolysis, net gain of ATP. First one, during glycolysis, 2 triose phosphate molecules are formed from 1 glucose molecule and 4 ATP molecules are produced. Out of 4 ATP molecules, 2 ATP molecules are utilized in first few steps in converting glucose to fructose 1,6-biphosphate. Moreover, 3 ATP molecules are produced from oxidation of each of 2 molecules of NADH produced during catabolism of glucose. In all, a net gain of 8 ATP molecules occurs during process of glycolysis. However, in anaerobic respiration, only 2 ATP molecules are produced. Krebs cycle is the second step of respiration and it occurs in the mitochondrial matrix. It was first proposed by H.A. Krebs. He was a British biochemist and explained how the pyruvic acid is broken down to carbon dioxide and water, and for this pioneer work, he was awarded the Nobel Prize in the year 1953. Citric acid is formed again and again in Krebs cycle, hence it is also known as citric acid cycle. A number of intermediate compounds in the cycle possesses 3 carboxyl groups, hence the cycle is also referred to as a tricarboxylic acid cycle. It requires oxygen. The pyruvic acid molecules formed during glycolysis are transported across the inner mitochondrial membrane and finally enters into the mitochondrial matrix. During this process, the pyruvic acid is converted into acetyl coenzyme A. It is a 2-carbon compound. At the same time, a molecule of NAD plus is reduced to NADH plus, H plus, and carbon dioxide is released. Acetyl coenzyme A now enters into the Krebs cycle. The reactions of the Krebs cycle is as follows. Let us study the one by one. First one is the condensation. The Krebs cycle shows the different steps involved. First one is the condensation. The acetyl coenzyme A combines with oxaloacetic acid. It is a 4-carbon compound to form citric acid. It is a 6-carbon compound in the presence of water. Then coenzyme A is released. Second step is the dehydration and hydration. The citric acid then loses a molecule of water to form cis A aconitic acid, which takes place up a molecule of water and forms isocitric acid. It is also known as isocitrate. Third step is the dehydrogenation. The isocitric acid is then converted into oxalosuccinic acid in the presence of isocitric acid and isocitric dehydrogenase. This reaction yields a molecule of NADH2. Fourth one is the decarboxylation. Oxalosuccinic acid is converted into alpha ketoglutaric acid. It is a 5-carbon compound by decarboxylation. During this process, CO2 is liberated. Fifth step involved in Krebs cycle is oxidative decarboxylation. Alpha ketoglutaric acid, it is a 5-carbon compound, is converted into succinic acid. It is a 4-carbon compound by using coenzyme A by oxidative decarboxylation. During this reaction, GTP and NADH2 are formed and CO2 is liberated. Finally, the removal of coenzyme A from succinic acid. Next step is oxidation of succinic acid. Succinic acid is a 4-carbon compound, is oxidized to fumaric acid. It is also a 4-carbon compound. During this reaction, FAD is reduced to FADS2. Next step is formation of malic acid. Fumaric acid, it is a 4-carbon compound, is converted into malic acid. It is also a 4-carbon compound by hydration. That means addition of water. The next step is oxidation of malic acid. Malic acid is oxidized to oxaloacetic acid. It is also a 4-carbon compound. During this reaction, NAD is reduced to NADH2. Oxaloacetic acid, it is a 4-carbon compound, again combines with acetyl coenzyme A. It is a 2-carbon compound to form citric acid. Citric acid is a 6-carbon compound. Thus, citric acid is repeatedly formed. Hence, due to the formation of citric acid again and again in Krebs cycle, hence it is referred to as citric acid cycle. The schematic representation of the Krebs cycle explain all these steps. Thus, the pyruvic acid during Krebs cycle yields about 4 NADH2, 1 FADH2, and 1 GTP, and liberate 3 molecules of carbon dioxide. Therefore, from one glucose molecule, which gives 2 pyruvate molecules, as a result, we get net profit as follows during Krebs cycle. Totally, the 8 NADH2 molecules are produced in 8, each NADH2 molecule here equals to 3 ATP. Therefore, it produces 24 ATP molecules. And during Krebs cycle, 2 FADH2 molecules are produced. The 1 FADH2 molecule is equal to 2 ATP. Therefore, totally 4 ATP molecules are produced. Then 2 GTP molecules are produced during Krebs cycle, and the 1 GTP molecule is equal to 1 ATP molecule. Therefore, 2 ATP molecules are produced during the Krebs cycle. And totally, there are 30 ATP molecules are produced during the Krebs cycle. The number of ATP molecules produced from Krebs cycle is about 30 ATP. Net gain of ATP from glycolysis is 8 ATP. And then here, out of 10, and 2 ATP molecules are used during glycolysis. Therefore, the net gain of the ATP from glycolysis is 8 ATP. Therefore, the totally 38 ATP molecules are produced by the breakdown of 1 molecule of glucose during aerobic respiration. Schematic representation of the Krebs cycle shows the different steps involved during Krebs cycle. Electron transport system. It is also known as terminal oxidation. It is the last step involved in the respiration. Here, It is a chain of hydrogen and electron acceptors embedded in the inner membrane of the mitochondria. ETS is the last step of the mechanism of aerobic respiration. It operates in the mitochondrial cristae, and 10 NADH2 and 2 FADH2 produced during glycolysis and Krebs cycle are oxidized in the ETS. When NADH2 is oxidized to NAD, and then FAD is reduced to FADH2. During this reaction, one ATP is synthesized. FADH2 is deoxidized to FAD and hydrogens are removed. The removed hydrogens then splits into protons and electrons. The protons enter the mitochondrial matrix and the electrons enter the electron transport system. The electron transport system is formed by the cytochrome enzymes which are arranged in specific sequence on the cristae, such as cytochrome B, cytochrome C, cytochrome A, and cytochrome A3. The two electrons are transformed from cytochrome B and cytochrome A3 by alternate oxidation and reduction of cytochrome enzymes. During the transfer of electrons, energy is released at two places and two ATP molecules are synthesized. At the end of two electrons are accepted by half molecule of oxygen and it gets ionized. Half molecule of oxygen is finally added in the protons and H2O is formed. In the ETS, ATP molecules are synthesized by the utilization of oxidative energy. This process is called oxidative phosphorylation. Image of electron transport system, how the ATP molecules are produced. Next one is the aerobic respiration, the process in which incomplete or partial oxidation of fuel molecules into ethyl alcohol, lactic acid, and carbon dioxide in the absence of oxygen is called anaerobic respiration. Anaerobe. What do you mean by anaerobe? Anaerobes are the microorganisms which function under anaerobic condition and breakdown of fuel molecules partially in the complete absence of oxygen molecule. Some plant physiologists use the word fermentation to describe anaerobic respiration. It occurs in bacteria, yeast, etc. What do you mean by the fermentation? Fermentation is a process in which chemical changes are brought about in the organic compounds by the action of the enzyme secreted by the microorganisms. There are two types of fermentations such as alcoholic fermentation and lactic acid fermentation. Let us we discuss about one by one. Alcoholic fermentation, the process in which yeast cells break down glucose into ethyl alcohol and carbon dioxide under anaerobic conditions are called alcoholic fermentation. Posture effect. What do you mean by posture effect? Sudden change from anaerobic to an aerobic process due to availability of oxygen is called posture effect. And the other definition of the posture effect is when the anaerobic yeast cells adapt to aerobic conditions in the presence of oxygen, the outcome is termed as posture effect. There are two types of the anaerobes. One is the obligatory anaerobes. Second one is the facultative anaerobes. What do you mean by obligatory anaerobes? Obligatory anaerobes are the organisms respires exclusively by the fermentation process and are killed by molecular oxygen. Next one is the facultative anaerobes. Facultative anaerobes are the organisms which break down their fuels either by aerobic respiration or by the fermentation depending upon the availability of the oxygen. Example, yeasts. Let us we know the lactic acid fermentation, the process in which breakdown of glucose into lactic acid in the absence of oxygen is called lactic acid fermentation. Last, I would like to take the respiratory quotient, that means RQ. The ratio of the volume of the CO2 liberated to the volume of the O2 consumed is called respiratory quotient. Another definition is the ratio of CO2 released to the O2 consumed during respiration is called the respiratory quotient. The RQ also may be defined as the ratio of the number of CO2 molecules released to the number of O2 molecules used during respiration. It can be calculated by using this formula, that is RQ is equal to volume of the CO2 liberated by, divided by volume of CO2 consumed. By this, I am completing bioenergetics, here in which the different steps involved in the respiration. I hope all of you understand the detailed account of the respiration. Thank you.